So today I'm going to talk about how to explore spatially resolved transcriptomes. And just to get a feeling, my background is actually from the left part here, it's technology. And then we try to apply that in sexy applications, but it kind of comes into the field of computational work as well, doing applied mathematics and also try to develop tools. I'm super uncomfortable to talk about the computational tools, just as a disclaimer for this talk, but I'll show some of the results that we obtained by doing this. And obviously it's exciting to be here since we had this ongoing collaboration with Rich and Himali and Silas and Tarmo, providing a chapter of ALS in this paper in Science that you probably have heard about many times already. But that's a beautiful example how you combine technology with an application and providing the new tools for computational analysis. So today's talks is focused a little bit on the technology just to get a start. And then I have two vignettes. One is actually on tumors, but I want to discuss a little bit of what we could do there. And then the second vignette would be can we annotate in a data-driven way tissues. And obviously you're well aware of the Human Cell Atlas, and there's a lot of activity here to also to build computational tools to address the Human Cell Atlas initiative. So I'm extremely fond of these kind of instruments. This is a DNA sequencer. It provides fantastic data when you like to measure gene abundances, the gene expression in tissues. It provides quantitative data, and you could either take your sample, put that into a tube, grind it down, extract the RNA, and use NGS technology to find which genes are present in that sample. More recently, obviously, you could take a tissue, you dissociate the tissue into single cells, and you could actually then enrich for the RNA profiles, the gene expression in those individual cells. Two very useful technologies. I'll come back to that. But what you should know already is that in the bulk analysis, you get an average view of gene expression from that sample, so you lose the spatial information. And that's also true for the single cells, although they provide very quantitative estimates of the single cells. And then you can look on the transcriptional profiles of those single cells, and you can cluster those into similarity clusters. So these are usually then denoted cell types because they share the same profile. But obviously there are a number of transient events between the different cell types that you analyze. So these are then signatures, but both of these technologies are then missing one important component, which is the spatial context. And I'll try to prove, and I think some of you already convinced that geography really matters. If you think about tumors, this is a prostate cancer. It has 7 loci, prostate cancer loci. And obviously you would like to know much more than the bulk analysis of this tumor to dissect out and understand what's the difference between these different 7 focal cancers. Another topic within the tumor field is obviously the interaction with the environment, the microenvironment of those tumors. What makes a difference between the different tumor nodes or loci here and their stroma? Can we find out interactions between these? So that's one important aspect. And obviously, if you think about atlasing, we have these beautiful images of brain where you have different morphological entities, and it would be super nice also to get the feeling of which genes are active to the different parts of the brain in more. I'll come into these topics, but from a genomics perspective then. So I'm representing something that we call spatially resolved transcriptomics. This is an expanding field. A lot of technologies is coming out continuously. What you could subcategorize these technologies is one is we use laser capture microdissection. You have a tissue, you isolate the cells that you would like to investigate, and you do RNA sequencing or genome sequencing of those. The second set of technologies is based on in situ hybridization. Since you know the genome sequence, you could design probes and throw these probes onto the tissue section and look where it hybridizes. And then you use clever fluorescent technologies to dissect out the different genes. And the third part is actually that on a tissue section, you're actually doing in situ sequencing. You could determine the sequence of genes. And again, there are a number of very good methods for that. So this is a long list. The focus here is really that these offers complementary information. And you have to consider then the resolution, the multiplexing capacity, and also the detection efficiency. That's the key markers. But what people kind of forget is that you have to have instrumentation, and you have to build up quite sophisticated computational infrastructure to get these technologies working. So in this context, we developed something that we call then spatial transcriptomics, which is addressing some of these issues. We love histology. We love the images of tissues because they are beautiful. And what you really do is just take a tissue, make a section, you stain it, and you get these H&E stains, and you could look for the morphology of that. So that's the image data. But then we combine it with this quantitative gene expression data provided by sequencing. And this is really done by a very simple trick, I would say. Usually when you have tissue and you section that onto glass surfaces, you could do that, but on our glass surfaces, we have a grid pattern. We have a spotted array with spatial barcodes. So you have, this is the tissue section, you position that on this grid, and each spot on the grid has a unique DNA sequence here in different colors. So we could then label RNA or barcode RNA from different parts of the tissue section. And then we use RNA sequencing to deduce the sequence of those genes, but also the position of those genes. And then we try to merge these two information sources to visualize the tissue context and the genes being present there. And I'll go through some examples. So this is a very short animation, if this works. Yes. So you could take any tissue in any organisms as long as it has a poly A tail. So this is a random tissue, and we do sectioning, slicing up the material that we're looking into. We usually use fresh frozen samples because you could extract the most of the RNA there. We stain it on the array. We take the image, which is one of the information sources that we like to, and we see the different structures, and we see the cells. And beneath the cells, we have this barcoding scheme. So each spot here corresponds to a unique DNA sequence, to a unique spatial barcode. So a side view of this is that you have a single layer of tissue, and you get the mRNA out migrating down to the surface and binding to that through poly-A hybridization. And then you perform a copying reaction of the cDNA, and then you remove the tissue, and you remove all the transcriptomes that you obtained from this entire section. And what we do then is always use these instrumentations to deduce which genes we have and their position. And this is traditional Illumina sequencing. So you're identifying the genes, but you also identify the position of that gene expression. And then you sit and try to merge these two information sources, and so you can look for your favorite gene or your favorite cluster of genes. And you could actually make a 3D model of the entire tissue. And obviously that's super interesting if you like to do atlasing, things like that. And I'll try to exemplify that. Some basic tech stuff. Well, how does this type of analysis look like if you compare it to doing in a bulk? Well, if you then make a proxy, well, if you combine all the spatial information into a bulk sample, you get this type of the on-chip data, and you go compare to a bulk, we could actually see a very good correlation over the gene expression space. So you capture both high abundant genes as well as low abundant genes. So we are not losing any genetic information by doing experiments on a glass surface. Another point is also that what we are doing here is just analyzing a part of the gene, the 3' end of the gene, which is something like 300 base pairs long. So these spatial barcodes, that's the secret to the technology. They have a mRNA capture region to capture the poly-A tail. It has a UMI to identify the unique molecules. You have a spatial barcode, and the spatial barcode then has different sequences depending on where you are on the glass surface. And then you have the handles to do sequencing. So this is then providing, what you practically do is that you order these probes and you let inkjet technology print these down on the glass surface. So you know exactly where each probe sequence is. And it's very repetitive, and it's the same kind of design every time you do an experiment. And we are developing, we have been developing these glass surfaces for some time, and the spot dimensions is going down. So we started out with 100 micron spots, the upper image, but now it's gone down to 55 micron. Well, actually 10x is providing 55 micron spots of genetic information. So recently there has been 2 publications that has tried to increase the resolution by using beads instead of a glass surface. And these beads have different dimensions. So in the first publication by Team at Broad, the size of the beads was 10 micron. And we, together with Rich and Tarmo, also have a recent paper where we use 2 micron beads. So you could actually capture the mRNA in a much finer detail. So what's the caveats of this? So the beads, you mix each bead with a probe, and then you distribute the beads in a random fashion onto the glass surface. So you don't really know what bead contains which DNA sequence. So you have to decode this before you do an experiment. And this is true for this slide-seq. You do in situ sequencing of the beads. In our case, we use the 2 micron beads. We use a hybridization scheme. And this is really a commercial way of doing it because we're using Illumina arrays for this purposes. So what is the possibility is then this is from the mouse olfactory bulb, and we could then generate much more fine structures of the tissue using these 2 micron beads. And this is a case from tumors, and you could see the landscape of different cell types within the tumor, and it's a pretty messy image. And I'll come back to that later on, because that's a challenge, I would say, from a biology point of view. So we have these kind of scenarios, and we have these very small beads or the spotted arrays. But what's clear from our experiments, obviously these are only pilot experiments in a way, the sensitivity is not really there for the things that we really like to do, which is data-driven analysis. So we are very fond of these other arrays, which have 10,000s of UMIs per measurement over a tissue space. So each spot then carries a few thousands of genes, and then you could select and look for your favorite gene, overlaying it with the histology. That's a low-hanging fruit. But I think the most exciting part is the data-driven analysis. What kind of patterns do we see in data? And we have some papers on bioRxiv, and Tarmo here is also developing these tools. And I think this is an area of great research activity, I would say. How can we adopt these spatial transcriptome-wide data? So I will then focus mostly on the data-driven analysis going forward. And this is just a demonstration of part of the brain. This is the most recent arrays from 10x, which you can see it's much more denser. And by computational means now, spinning out from the bioRxiv of the probabilistic methods that we're using, the data looks something like this today, which is looking very nice, I would say. Another aspect that I would like to also take advantage of is the spatial data actually has a very nice counterpart in the single cell world. And there's a lot of effort now to then within the Human Cell Atlas to characterize all human tissues by the single cell RNA sequencing. But the important thing is it's a similarity from a technology point of view, we're always looking at the same part of an mRNA species, the 3' end. And the correlation between two different methods look very, very good, actually. And that's something that we could take advantage of when we are designing, when we combine technologies. So mapping, so we could then do our ST experiments, and this is then from some tumors. This is HER2 breast cancer tumors. I'm not focusing on any details here. This is triplicates of a number of patients and their tumors. We generate the ST data, but then we could link into the single-cell data sets that has described how adipocytes look like at the gene expression level, or the CD8 cells, or the macrophages and CD8 cells. So we could actually find, combining these two, where the different cells are. A challenge, I think, as I mentioned earlier on, is tumors. It's extremely challenging because you have large patient-to-patient variation. And we definitely need new tools here to better understand this. So this is just an example. These are then where we have triplicates of 4 patients. We're looking for T cells. Where are they present? And the more blue indicates of presence of those cells. And you could actually see that a very wide variety of presence of these cells. And this is much work to do. And I'll come back to this later on, actually. So if we just take the first vignette, and this was the story of technology and how can we actually benefit, how can we use this type of data. And one of the first experiments was from cancers. And this is a work that we did many years ago using bulk RNA sequencing. In this case, we have 4 biopsies from 4 patients, and they all had metastasis in the lymph nodes. One of these patients was a long-term survival. The 3 ones was short-term survival. So we have some outcome data. And we were super excited like 8 years ago when we get those data, because this is an hierarchical clustering of the most differentially expressed genes, and we could actually see good reproducibility between the 2 samples. And that's good, but we could also see that each patient has a unique signature. We're super thrilled of that, but actually when we took those samples into the spatial perspective, a completely different view was found. So this is really what that we work with. This is H&E stains. Pathologist is annotating this as tumor. This is lymphoid tissue, and this is stroma, and this is then stroma and lymphoid tissue as well here. And we put these onto the arrays, and we do some data-driven analysis. And now generating clusters that share the same gene transcript profile per spot or tissue domain. And this is how that could look like. So you could actually then look, as a positive control, you could look for lymphoid tissue here. And you see that it's a light blue color, and you actually could see that the same light blue in both samples, which is a good feeling. We are on track here. But if you look on this tumor, you have one gene transcription profile here indicated in violet, while this tumor has multiple gene expression profiles. And you would not have seen this by bulk analysis, definitely, and you would not probably understand this complexity by single-cell RNA sequencing. So this is a beautiful example where spatial components actually add information to this. And obviously, if you think about digital pathology, you would never find this kind of patterns if you haven't looked into the gene expression profile. The histology in itself is not sufficient. So this kind of led to that we would like to embark on larger studies of tumors, and this is one of the first studies on prostate cancer, where we analyzed some 6,000 tissue domains within the tumor. So this is a whole prostate here, and this is the legend for we have multiple places where we have cancer, we have normal glands inflammation, and yet another way of cancer. This is the severity of cancer as in terms of Gleason score. We put these onto the arrays. Again, we do some machine learning. And we could then collaborate with the pathologist who's indicating this is tumor. Data-driven way indicates, well, it is indeed a cancer profile, but the area is much larger, not evident by microscopy. And we could confirm that by immunohistochemistry, that the area actually is larger from that perspective. So we believe that this is actually a very useful clinical tool because it will aid the pathologist to annotate what he thinks is cancer. Yes, it's a cancer profile. The second one, he suspected this was cancer. We could confirm that, and we could actually find also areas that has a cancer profile that was not picked up. So we see that machine learning will have an impact also with this type of studies. And I had a presentation at the European Society for Pathologists the other week. And after my presentation of this, I'm very biased. I think this is super sexy. He said, well, these modern technologies will never impact pathologists. So he likes to look at the H&E stain only. That's a take home from that lecture. Anyway, still we are continuing this kind of dictionarying of tumors, so we could have a profile for normal stromal cancer and so forth. But as for the melanoma, it's actually more complicated, right? So this is a prostate tumor, and we get, this tumor has 3 different signatures, and you could see that these are spatially separated. So we are not determining only one tumor, but also the variants of a tumor by its gene expression. So this is then appropriate for lunch. This is a prostate cancer where we try to take this to get a global image of the entire tissue. And this is ongoing work, but we are actually sectioning the tumor here. And we have a 2-dimensional grid, but also a 3rd-dimensional analysis ongoing for this sample. And just want to share a glimpse of this. This is data-driven annotation of the prostate as a whole. We analyze some 20,000 tissue domains. We identify some 7 cancer expression, and we see these gradients that I talked about early on, which is super important to understand the gradients around the tumor. And we could clearly see that we have gradients here. Yeah, so this is an example, maybe it doesn't come across clearly, but you have cancer here, and you have a color gradient here close to the cancer. This is normal epithelial. You have inflammation. You have a lot of things, events appearing in the tumors that are nice to catch, actually. So this is also another example how we merge spatial data with single-cell dictionaries. And this is on bioRxiv. And I'm just going to give some glimpse of this. Again, this is the H&E stain of a tumor. This is invasive cancer. The dimensions here is 5 by 5 millimeter. And we look for a cancer profile. And to make certain that we are on track here, let's see if this works. I don't see my... Here we go. Maybe. That was a very strange movie, actually. So we are sectioning the tumor multiple times to understand that what we see is actually true. And we see a cancer profile on this corner correspond to this part. We have no cancer profile here. So having these dictionaries, we could ask this tissue sample, do we see any aberrations in terms of the immune response, immune cells? And then we ask, do we see cytotoxic T cells? And you should know that if you have infiltrating cytotoxic T cells in the tumor, that's a good sign for the treatment for this type of cancer. So you could actually see, which I think is kind of striking, that this tumor is so-called cold, meaning no infiltration, while this tumor on the other side, coming around the corner here, down here, has infiltration of immune cells, which I think is super interesting because it kind of proves that we have a complex thing with the tumors here. in terms of composition of different immune components in terms of the cell.
Is that color just CD8? Or is
Yes, that's only CD8.
Okay.
Yeah. Yes. So something that obviously is important, which we did also in the ALS story, was to think about time. How could that affect gene expression changes? And we have one example from this, from the tumor world, which I will just go through shortly. So if you get prostate cancer, you identify it, you have a tumor volume here, and over time these will grow. And if you identify it, you usually get so-called ADT treatment, which stands for androgen deprivation therapy. So androgen is what drives the tumor to grow, and what you really try to block this expression. So you start this blockade, and the tumor shrinks, but at some point resistance will occur over here. And overly at some point you cannot treat it anymore. So the dogma in the field is really this, that you have a tumor, you treat it with ADT, and these colors then corresponds to tumors that respond. These are then stroma cells that are important for tumor growth, and that you are actually eliminating the green ones, and the resistant tumors occur through treatment. That's the dogma, right? But we could actually then try to address this in a spatial way. So what you really take is needle biopsies here from a group of patients. This is a core needle biopsy throughout, and we are staining for this androgen receptor here. And you could actually see that they're staining. So this is, then you could compare these needles before and after treatment. And what you see here, this is a responder. So this is the intensity along this core needle biopsy, and after treatment, it's actually gone down significantly. So that's a proof that resistance, that you have a response to this treatment. But when we go over many patients, you actually see much variable results. So some don't respond at all, some do respond, and so forth. So you have a mixture of the signal. So what we've done here then is to use the pathologies. We do this data-driven ST analysis, and we then color codes the different signatures. So we have, in this case, for this small needle biopsies, we have 4 different cancer clones, we have 2 different stroma clones, and we have 3 immune clones. And when I say clones, I mean expression clones and nothing else. So we could actually monitor how these different components of a sample varies over time. So this is before treatment, you have a cancer here, and the pathologist confirms that it has red color here, and if you just look for this factor in this case, it's positioned there. And then after treatment, you could take a new biopsy. Obviously, it will not be the same position, but you look for these red ones, and in none of the biopsies that we take, we could find these cancer clones. So this is really a responder, similar to the one that I showed before. But in this case, we have a tumor here. It's annotated to be tumor here in this color, turquoise. It's located here. But after treatment, we actually see this turquoise also present here. And we could confirm that also in here by immunohistochemistry. So the same clone that was before treatment is still residing here. So what is that telling us? Well, that tells us that we most probably have a new way of looking at tumor resistance. So it kind of indicates to us, this type of data, that we have the tumor cells here, but it also encodes the resistant clones already before treatment. And this is really, really good news, because then you could take a sample before treatment to predict whether or not this patient will develop resistance. We are super excited about this data, actually. The last vignette is the data-driven annotation of tissues, or atlasing, and this is something that we put on bioRxiv quite recently. It's can we annotate tissue in a data-driven way? And the example comes from the brain, which has been studied over 100 years, more than that, from histology point of view. But the Allen brain obviously has put a lot of effort into getting a histological structure of the brain with some connectivity underlying the different areas of the brain. And this we've done together with Dinos and Cantina Karolinska Institute, who has a long-term interest in the brain. So what we do is very similar to what we did in ALS in a way. We have our histology, we have our spatial data, we make these annotational regions, we do our measurements, and then we apply registration. So we put spatial data into this coordinated framework of the brain. And the framework was published some time ago in Nature Neuroscience. So what we have here then is the mouse brain. We used 3 animals, 75 sections. These are the different animals, and these are the sections through the brain. And we're analyzing some 34,000 tissue domains, or spots. So in this data, we have about 4,000 genes per spot, per measurement, and we characterize a majority of the genes present in the mouse genome, something like 15,000 genes. So we take advantage of the Allen Brain Initiative by putting our spatial data into a coordinate system of Allen Brain. So the isocortex here has about 10,000 measurements and so forth. And we are actually able then to get we are able to get DNA data or gene expression data from all of these regions that Allen has defined. So all in all, we are throwing around the transcriptome wide for 34,000 different positions within the brain. So the low-hanging fruit obviously is to look for clear markers for the different regions. So here you have the cortical marker. This is a thalamus marker. This is a striatal marker, and so forth. So this is generating data. You could just cherry pick your favorite genes, and you could position these in a 3-dimensional framework and look for that. But obviously, the data-driven way of doing it, it's even more exciting because we see patterns that has not been seen before. So this is some 181 molecular clusters, but as you know, you could choose another numbers, but this was functionally, these were spatial meaningful data. So we use ICA for this purpose, and we get patterns then that we could see that on a 2D perspective looks realistic or good, and in 3D it makes even more sense, and we could do this like that. But we're also able then to identify technical clusters that is only present in one section, and we could remove those, we could regress out those data. So here we have this cloud again, but here we're using the same cloud by using the measurements from the Allen brain. Green was the cortex part of the brain, and you could see all these make sense. So there are some structure also in this ICA analysis. Yes?
So each dot represents a location in the brain and is clustered based on the gene expression.
True.
Location.
Exactly.
And what does the color represent?
This is a random color, but this is the colors used by the Allen Brain Initiative.
So the color on the right maps in the color of the little diagram.
Exactly.
And the color on the left is
Just random color from clustering. So here you could actually see then how we are able to build up the sheets of gene expressions along the structure of the brain. So this is actually the first time that the brain then will be molecularly defined, unbiased of human interpretation of the brain structure. And you could see actually how it makes sense. So this is a very, very long movie since we are building up the entire brain, so I will not show all of it, but you get the feeling of how they superimpose over each other. So if we look for this one, which is this part of the brain, this is the Allen brain. You have structures here, but in this part, which is the striatum, you don't really have any morphology. You have no structure. So we have then collected the data from this part, and it's hard to see, but we are connecting them, the different measurements by support vector machines to get these sheets that I just talked about. And this is the outcome from this section. And you could actually see that this histologically, morphology, non-informative region actually contains a number of new regions defined by gene expression. Does this make sense? Well, if you make this type of similarity analysis, we have the Allen brain annotation on the outside here, and inside we have the molecular annotation. And if you look for striatum that I just showed, which is light blue, you have all of these subclusters of gene expression, and you can see that these actually make sense. So we're actually able to increase the resolution of the brain using these data-driven approaches. Yeah, I don't really follow what you mean.
Sometimes you see some expression patterns that kind of cross from the cortex to certain, like hippocampus, some RNAs kind of expressing, not necessarily strictly bounded by the regional.
Yeah, I think in many of those connectivity things, you have to have a higher density of the measurement to be able to pin down a single neuron that connects different parts of the brain. So obviously that's would be fantastic. As I said early on, the sensitivity of these subcellular arrays are not there yet. So that will only look for the biomarkers, the top biomarkers. But I think that's a super good potential to do that in the future. And this is, oops, it started here, oops. Let's stop here. So this is actually then a movie, how we look throughout the brain. And to the right, we have the Allen brain annotations, and to the left is the data-driven way. This gives a little bit of a three-dimensional feeling. Let's see if this goes. And you could actually compare them, previous annotations to the right, the data-driven way. And you could see a lot of similarities, but you could also see a lot of more resolution in the analysis of this. And this is obviously a fantastic tool then to analyze our organs in 3D and just using the molecular information for that. And that's a property that we have used in the last vignette, where we try to use these type of tools to understand the development of 3 human organs, which is brain, heart, and lung. And this is part of the Human Cell Atlas initiative, and we have a Swedish team. And I'm a technologist, so we have a fantastic set of technologies at Scilab Lab, providing different measurements into the same organ. I think this is a fantastic tool for you guys as computational, because this is will be a very rich resource. So we have Emma Lundberg doing proteomics using Gary Nolan technology of Codex. We have myself doing spatial. We have Mats Nilsson that is doing in situ sequencing that I mentioned early on. Sten Linnarsson is doing single cells. We have tissue provided by Erik Sundström, and Christus is doing some fish analysis. So what we have done, this is ongoing work where we have collected single cells from brain, lung, and heart. These are clusters, but this is just pinpointing where we have lung, heart in this case. This is the reclustering of the heart and looks good. So having this resource, we could go into spatial analysis and of different types, both protein and RNA. So practically what we do is that we are sectioning then our samples, which are extremely precious, obviously, for the different technologies, ST, combining that with in situ sequencing and doing proteomics on this. And combining that with some data-driven analysis of single cells. And together, we are creating a very rich resource, and this was recently accepted as a resource in Cell. So what we can then pinpoint is how the heart is developing over time. And we use these spatial patterns and the single cell to pinpoint the cell types in the different parts of the heart. So this is an organ-wide description of all the genes being important in that development. And that is meeting a big medical need for the issues in newborns. So again, we are generating these kind of three-dimensional maps of different anatomical regions. We are then using the spatial data to generate these maps, and this is super exciting. And the possibility to combine technology on the same stacked technologies is very useful. These are then 5 genes representing different parts. This is single cell data, and these are the marker genes for different parts. And we could then position these spatially by ST. This is the low resolution arrays, but you could actually see that this is atria, combines atria. This is ventricular, looks like that. This is the outflow where the cells are, and this is some epicardial markers. So then finally we could take all of that data and confirm that by these panels. So this is in situ sequencing, confirming what we have from single cell and ST, and confirm that these are truly the markers that pinpoint different anatomical regions. So we get a very high resolution map of development. So that was my end image. I hope I kind of convinced you that geography is really important and that we could use these type of data-driven approaches, and there's plenty of room to improve the annotation of tissues, annotation of tissue sections, but we could use these tools also to pin down different disease mechanisms in a very non-biased manner. I think actually the wealth of information provided in single cell the field is very useful, because then you could map those cells into different aspects of brain development or in tumors. I particularly like this temporal analysis, which was explored in the previous ALS study, is to see how spatially things change over time. I think that's super, super sexy. And with that, I should also say that we are a team of 3 PIs doing this work, and I try to pinpoint who's contributed to different parts throughout the presentation. Worthwhile of noting is Sanja, obviously, that's done the ALS and also the high-density arrays, and all the set of collaborators we have, including you guys that are sitting here, and the funders for the research being done. Okay, with that, I am open for questions, and I would like to thank Rich for providing this Swedish fish in my hotel room. That was super clever, actually. Yes, this is important to feel welcome. So, thank you.
Use your seat mic, please.
Can you do immunohistochemistry on the same slide that you apply spatial technology, so that maybe you can look at diffuse growth factors and so far in association with the transcriptomes?
That's a super good question. So H&E staining works just fine. Certain antibodies that works very efficient, where you have a short incubation time, they will work. The majority of antibodies needs an overnight incubation or something like that, then the RNA will be degraded. But I think without pushing this too much, you will find ways to actually do combine these measurements much better. So for instance, we have used from the side-seq kind of antibodies, which has also a barcode. We combine those on our arrays, and that looks pretty good, actually. Okay.
Combining with the proteomics, when you're doing, say, like the heart, what are the relative considerations with the proteomics data? Is that like an emerging technology, or I'm not as familiar with that, or is it dense enough that we can actually do kind of straightforward comparisons with the ST?
Yeah, so that's a good question. The proteomics has, it's like they can only look for a short fraction of all the proteins present. It's not highly multiplex, so you could look into 30 or 50 or so proteins at the same time. And there are tricks around that in terms of fluorescence and in terms of antibodies that you use and stuff like that. However, I would say that we've done single cell in collaboration with Australia on single cells. You have these clusters that are driven by RNA profiles, but then we use this kind of side-seq on top of that, and that improves the cell type annotation dramatically. So we have a much better view of the clusters by incorporating the protein information. So I think there's a future for proteomics, but it's not very multiplexable so far. Okay. Thank you.